Welcome back. This is going to be the last video for biochemicals. We're going to focus specifically on enzymes. Now, what are enzymes? Enzymes are special proteins that speed up chemical reactions. And things that speed up chemical reactions, we call them catalysts. Or we use the word catalyze. They catalyze the reaction. And I'm going to get into this in the next slide of how they catalyze the reaction, or what a catalyst actually does. So enzymes are also referred to as biological or organic catalysts. Enzymes help molecules combine or break apart. Depends on what you're trying to do. Without the help of enzymes, many reactions can't happen inside living organisms. Why are enzymes so important? Enzymes help reactions happen by lowering the amount of energy required to run the reaction. What does that mean? This makes the reaction more likely to happen and easier to happen. So if you're looking at this diagram right here on the bottom, you have this red line. This red line represents actually the chemical reaction happening with an enzyme. So you see how this energy level is much lower than where it would have been without an enzyme. So the activation energy is how much energy is required to get things rolling. So think of it as some mornings it's harder to get up in the morning than other mornings, and maybe the enzyme would be your parent or guardian coming into your room and getting you out of bed that much quicker versus doing it on your own. Just to put it into perspective of what I'm trying to explain. But essentially, they help the reaction happen faster and easier with less energy needed by the body. So how are enzymes named? Names of enzymes usually end with ACE, ASE. So protein enzymes may be protease versus lipid enzymes, which are lipase, and maltose, which is a type of sugar, will be maltase. Okay, I hope you got it by then. It usually ends in ACE. Enzyme vocabulary, you have your substrate, which is the molecule changed by the enzyme. You have your active site, which is the part of the enzyme in contact with the substrate. And the enzyme and substrate have specific shapes that fit together to make an enzyme substrate complex. So looking down at this picture, you have your substrate. This is the thing that is actually going to be broken down later on. Your active site is where that substrate is going to bond to the actual enzyme. And if you look in the middle, you're going to see that the enzyme bound to the substrate at the active site. And after the enzyme did its job, it's going to break that substrate into two subunits. Doesn't always have to be break, but it can be. Another example, you have this green blob, which is the enzyme, and you have this free substrate that's going to bond at the active site. So now moving down to that bottom left image where it actually bonded together, and moving over to the right, see the enzyme actually did its job by breaking that substrate into a smaller unit, and then it's released. Just want you to notice, did anything happen to the actual enzyme in both this picture and this picture? So bringing you back to the idea that I was just asking, are enzymes reusable? An enzyme only helps out, so it's not being used up. Okay, so it does not change chemically during the reaction. So as a helper, it can be reused over and over and over. It really just comes down to how much enzyme do you actually have and how many substrates do you have that need to get broken down or put together. So what factors can affect the reaction rate of enzymes? You have the amount of enzyme present, you have the amount of substrate present, you also have temperature and the pH level. All of these things affect how well your enzyme will work. So what conditions are optimal for enzyme function? You need to have optimal temperature and pH that the enzyme likes, and that will change per enzyme being used. Also, certain organisms have different enzymes that function at much higher temperatures versus lower temperatures. In humans, 37 degrees Celsius is normal body temperature, whereas in Fahrenheit it's 98.6. They're both equivalent. So our human enzymes will most likely be optimal at those two temperatures, 98.6 Fahrenheit or 37 degrees Celsius. Now, This depends on specific types of enzymes, like I was just saying. So if you're looking to your image to the right, now you see how the enzyme activity is going to increase all the way until you get to 40 degrees Celsius with this enzyme. But what happens? Why is enzyme activity going down after 40 degrees? We're going to talk about that in just a little bit. But I want to focus your attention to typical enzyme graphs. So in the top graph, you have a human enzyme compared to a thermophilic, and thermo meaning heat and philic meaning liking or enjoying. So thermophilic bacteria. So that means they thrive in very hot conditions. So if you're looking at this left hump, it actually ends up being optimal at about 35 degrees Celsius versus this thermophilic bacteria enzyme, which is more optimal at 70 degrees Celsius. Now, humans would be dead at 70 degrees Celsius. That's a very high temperature. To put it into perspective, it's about 158 degrees Fahrenheit. That's extremely hot. That's almost, what, shy of 50 degrees from boiling point. That'd be really, really hot. Now, the bottom graph is actually representing two human enzymes affected by pH level and the rate of reaction. So on the left-hand side, you have pepsin, which is actually found in your stomach, and its optimal pH that it's functioning under is just below 2, which is on the acidic side. And if we think about our stomachs have hydrochloric acid, so they're on the acidic side. That's where they're going to be optimal versus the other object, which is trypsin here, that can be found in other locations of your body, is more optimal around 8, which is more on the basic side. So all things can be affected by different pHs or different temperature. It's just about having that optimal pH and optimal temperature. So what happens if the conditions are wrong? Remember when I was going back to this graph right here and I asked you what happens after 40 degrees? Now we're going to talk about it. The shape of the enzyme may change, and we call that denaturization, or the enzyme is denaturing, which results in a poor fit of the substrate. Now, if the enzyme and the substrate don't fit perfectly, it's not going to work. Nothing's going to happen. The enzyme can't do its job. So if you raise that temperature too high and you change the shape of the enzyme, you no longer have anything functioning. Denaturization occurs if the pH is out of range for the enzyme or the temperature is too high. But if the temperature is too low, enzyme activity just slows down. It does not denature. Nothing is affected. Now, enzymes, we use enzymes for chemical digestion within food vacuoles of a cell or within human digestive systems. Lysosomes also have digestive enzymes inside them. Now, all metabolic reactions actually use enzymes to speed chemical reactions on in the body. And later on in the year, when we talk about DNA electrophoresis, we're going to talk about using enzymes to cut DNA in specific locations so that way we can run them through a gel and run them in different sized fragments. I do want to draw your attention to a practice problem. I'll read it for you. I want you to pause before I go over the answer and try it on your own. But simply, the question is asking, state one valid conclusion that relates enzyme concentration to its reaction rate. So I'm going to read it, and I want you to pause after I'm done reading and try and answer this. An investigation was carried out to measure the rate of activity of catalase, an enzyme that breaks down hydrogen peroxide. 5 40-milliliter solutions of the enzyme at concentrations of 20%, 40%, 60%, 80%, and 100% were prepared. A filter paper disc was placed in each enzyme solution. Each soaked disc from the different enzyme concentration was then added to different cups containing 30 milliliters of 1% hydrogen peroxide. The rate of catalase activity was inferred from the measurements of how fast the discs rose from the bottom to the top of each cup. The following data were obtained. 40% solution took 12.1 seconds, 80% solution took 5.8 seconds, 100% solution took 4.1 seconds, and 20% solution took 15.8 seconds, and the 60% solution took 9.9 seconds. So state one valid conclusion that relates enzyme concentration to reaction rate. So please pause the video and try this on your own. Hopefully you had a second to pause the video and try this on your own, and you came up with the conclusion that as the concentration levels of enzyme went up, the time it took the rate of catalyzed activity went down. Okay? So that would be one valid conclusion that you could have made. I hope this helps and watch this as much as you need. If you have any questions, please come to class with them.